Hi everyone, I'm Tian from the University of Melbourne in Australia. I'm very glad today that I'm here to chat with everyone about the two most commonly used models in representing brain organization, the smooth gradients and discrete regions, or a bit of both. Okay, let's start. So the brain organization has been studied for over a century. We believe that the structure, function, and connectivity of the brain are topographically organized like a map, in contrast to a diffused or random phenomenon. So classic models of brain topography parcellate the brain into distinct partitions with discrete boundaries, while recent studies of cortical gradients suggest that the topographic variation in brain structure, function, and connectivity might be better conceptualized in terms of continuous gradients of gradual change in the absence of discrete boundaries. So this new ideas of brain, this new idea challenges the traditional notion of brain organization and gives rise to the contention about whether the brain is organized into distinct partitions or continuous gradients. So here are the two models of brain topography. One suggests distinct cortical patches. One suggests continuous gradients of gradual change. So are the two models contradictory? Can we somehow reconcile these two models of brain topography to represent the brain organization? So of course, on one hand, we can continuously characterize the spatial variation in brain structure, function, and connectivity across the topography. On the other hand, we should also be able to delineate a hard boundary when the spatial variation is significantly large. So how large or how significant the spatial variation must be to warrant such a hard boundary? We conceptualize such a question as mode selection in our work. So in today's talk, I will briefly introduce principled approaches to reconcile these two complementary models of brain topography. In particular, I will introduce the idea of mode selection in the context of gradient mapping in two brain areas. One is the insular cortex and another one is the subcortex. So here is the insular cortex. The insular cortex is traditionally and most commonly categorized and parcellated into distinct subregions. Each subregion presumably represents distinct patterns of structure, function, or connectivity. However, as you can see the few examples I show here, the number of insular subregions and the subregional borders differ substantially across different parcellation studies and parcellations. And modalities, sorry. So to understand the high inconsistency, we systematically evaluated traditional clustering approaches in insular parcellation and the new models of continuous gradients. So we found that the topographic variation in insular functional connectivity is actually best represented as a continuum along an anterior-posterior axis. So as you can see here, the functional connectivity profile varies at a relatively constant rate from the posterior to anterior, while the slope changes a little bit at breakpoint 1 and 2. The evidence of indicating a sharp boundary in this continuum is relatively weak. So this may in turn suggest or explain the high discordance in the number of insular subregions that previously reported. And moreover, so this continuum is also consistent across several imaging modalities and early work of track tracing in non-human primates. This also suggests the structural function convergence in the insular topography. So now how about the subcortex? We have characterized that the insular topography is best represented as a continuum. So I now want to apply the model selection framework to investigate the subcortex. The subcortex comprises multiple structures and are widely implicated in the pathology of many neuropsychiatric disorders. However, many of the structures have been studied in isolation without first establishing the functional boundaries between them. The very few existing connectivity-based subcortical atlases, including more than one subcortical nuclei, generally do not preserve such anatomical boundaries. So to understand whether this is a structure-function divergence, in contrast to the convergent phenomenon as we just see in the insular cortex, we use the idea of cortical gradients to continuously map the spatial variation in functional connectivity across the topography of the entire subcortex. So the idea is that we would only delineate a boundary when the spatial variation is significant and is justified by the model selection process. So before I move on to show you the results, I would like to provide a brief overview of how connectivity gradients are typically mapped, given that this is a relatively new concept in this field. I will describe in the mapping in terms of the functional connectivity in the subcortex. So here I show that we first map the functional connectivity between each subcortical voxel and all the other gray mapped voxels in the brain. And then we quantify the similarity in the connectivity profile, in the whole brain connectivity profile between each pair of subcortical voxels. So here, instead of using the traditional clustering approaches to group voxels into distinct clusters, as traditional postulation would do, we decompose the high dimension similarity matrix into a few low dimensions using one of the eigendecomposition methods. So the method we use is called Laplacian eigenmaps, and each of the resulted eigenvector represents a dimension of connectivity difference in the subcortex. Those eigenvectors are typically and normally ordered accordingly. So the first eigenvector or the first gradient explains the most variance, while the second explains the second most variance, etc. So to simplify the explanation, today I would only focus on the first connectivity gradient, which explains the most dominant connectivity difference across the topography of the subcortex. So here we have the eigenmap, the dominant gradient map. So we want to further characterize the connectivity gradient, we developed a technique called gradientography. Let's see how it works. So based on the eigenmap, we then estimate the gradient orientation for each subcortical voxel in the eigenmap, such that we can fit a tensor into the gradient, into the gradient field. Why we need tensor? So after we fit tensor, the shape along the long axis of each tensor represent the direction of the gradient, whereas the gradient magnitude is characterized by the length of the tensor. What we can do then, so as long as we have the tensor, what we can do is to propagate millions of streamlines in this tensor field in a similar way as white matter tractography is typically performed in diffusion imaging. But here we propose the tractography that is based on the connectivity gradients, so we call that gradientography. Let's have a look. So those stringline propagate, let's have a look at those propagated stringlines. Each stringline represents a trajectory of maximum change in the eigenmap. So here we color the stringlines based on the gradient magnitude of each subcortical voxel. Locations with this kind of circumscribed bands here, here, or here indicate the local maximum in the gradient magnitude, which suggests a sharp transition in the connectivity, and also in turn suggests the functional boundaries. So the idea of gradientography enables not only a striking visualization of the brain gradients, but also the identification of sharp transition, which is the functional boundary. So let's take a closer look. Here are the stringlines that are traveling along the basal ganglia, and we can see transitions between globus pallidus, putamen, nucleus cumbens, and the caudate. We can then parameterize such transitions in a two-dimensional space, which is the idea of diversity curves. And we can see the three transitions, the peaks in the diversity curves that corresponding to the locations of the transitions. Similarly, we can do the same thing for the ventral part, and we see that the stringlines that go through the left, right thalamus, hippocampus, and amygdala. Similarly, so more local peaks are noticed in the ventral part of the diversity curves. So now the question is, shall we consider all the peaks? To what extent the peaks or the transition is driven by the matter of chance or other confounds, such as the extent of spatial smoothing during the fMRI preprocessing and also the geometric contraction in the subcortex. So to control those factors, we generated a null model as, as the gray curve show here. So the idea is that we parcellate the subcortex at locations when the empirical peaks are significantly larger than the peaks in the are significantly larger than the null curve, such that the null model can be rejected. Otherwise, we argue that the topographic variation in functional connectivity is best represented as a continuum in the absence of discrete boundary. So this model selection process yielded an initial parcellation for the subcortex with 8 bilateral comprising 8 bilateral regions, recapitulating a well-known anatomical boundary, anatomical nuclei. So it will be 16 in total left and right. We then map the connectivity gradient for each of the 16 regions with the performing of model selection recursively. This yielded a hierarchical or multi-scale subcortical parcellation that comprised of 4 organizational scales. But it's not, well, I want to say that it's not all the subcortical regions comprise 4 scales. As you can see here, the globus pallidus or the amygdala only comprise 2 scales. In contrast, hippocampus and the thalamus extend further into scale 4. And this is consistent, so we think that this is consistent with the greater functional and organizational complexity in the hippocampus and thalamus compared to the more specific role of globus pallidus in the movement control, regulation of movement control. So this multi-scale subcortical atlas is free to download. I'll show the link later. And this atlas has been utilized in ongoing clinical trials of brain stimulation like TMS and DBS in the treatment of patients with neuropsychiatric disorders, which is very promising. And this slide, I want to show some results that is a little bit beyond the scope of today's talk, but it's quite interesting, so I would like to share. So far, we have only investigated the functional connectivity gradients at rest. So we are wondering whether the connectivity gradients reconfigures in response to changing cognitive demands, which means in the task-induced conditions. So to answer this question, we mapped the diversity curves for 7 task fMRI conditions, including, as you can see here, emotion recognition, gambling, language, modal execution, relational, social influence, and working memory. So those 2 curves, the red and black, are mapped from the resting state, whereas the other seven curves were mapped from the task fMRI data. So indeed, we found that the subcortical topography shows reconfiguration in all 7 tasks compared to the rest, as you can see here. So in particular, during the task conditions, as I pointed, the anterior putamen integrated more extensively with caudate and nucleus accumbens that are associated with higher order cognition. In contrast, when we look at the ventral part, the hippocampus segregated more along its longitudinal axis, yielding a more integrated anterior hippocampal amygdala system for emotional, maybe have served better for emotional regulation, and more integrated posterior thalamus that may have benefited learning and spatial memory. So this dynamic reconfiguration may actually reflect a process of neural recruitment into specific functional processing systems, so to cope with those external complex cognitive demands. So to wrap it up, we propose a model selection framework. So this framework can can reconcile these two representations of brain topography. It enables the disambiguous, or we disambiguate smooth connectivity gradients from discrete area boundaries. So we show evidence while so the insular cortex, the functional connectivity topography of the insular cortex is best represented as a continuum in contrast to distinct pulses, whereas the subcortex has a more complex structure, organizational structure. So we proposed a hybrid representation incorporating both continuous gradients and distinct pulses. So the multi-scale subcortical atlas. So here is the link for the atlas downloading. We give the name the Melbourne Subcortex Atlas. So please feel free to download and use in your connector mapping and whatever study you are interested. And if you are also, and also here, check out this preprint if you are also interested to know how subcortical topography relates to the intrinsic organization of the cortical networks and how subcortical functional connectivity explains variations in reward and goal-directed behaviors. Yeah, I don't have time to discuss them further, but if you are interested, please go to this preprint. There are more details there. And finally, I would like to thank everyone being involved in this project, including my supervisor, PhD supervisor, Andrew Zalaski, and other collaborators, Michael and Daniel, and all the other friends. And particularly, I would like to thank everyone who has been listening. And I think it's time for questions. Any questions? Thanks.